Today we will discuss monohybrid. Inheritance. Monohybrid inheritance is the study of transmission of one characteristic from parents to the offsprings. Okay, so one gene is inherited from parents to offsprings. So this is monohybrid inheritance. Okay, we have dihybrid inheritance, where we will study inheritance of two genes together. And let's see monohybrid inheritance. There are four modes of inheritance. It is complete dominance. It is codominance. Incomplete dominance. And we have sex-linked inheritance, or sex linkage. So this is autosomal inheritance. So let's see complete dominance. Complete dominance. If a pure breed plant for round seeds this is pure breed round seeds is cross-pollinated with another plant pure breed for wrinkled seeds. The offsprings produced is with all round seeds. Okay, and is known as an F1. When two pure breeds are crossed together, the offsprings produced is known as an F1. F1 stands for first filial generation. Okay, so in the F1 generation, one parental characteristic expresses itself. One parent had round seeds and other parent had wrinkled seeds, but all the offsprings produced have round seed. So a characteristic which expresses itself in F1 is known as a dominant character. Okay, and it is due to a dominant allele. Dominant allele is represented with a capital letter. So we'll come back to this. Let's see. We have discussed pure breed. So what does a pure breed mean? An offspring or an individual? An offsprings or an organism? Which has both of its parents of same phenotype and if it is crossed or self-crossed, it produces offsprings with its own type of phenotype. Or simply we can say a pure breed is homozygous. Genotype. Okay. So if this is, for example, a round parent and it is self-crossed with round parent, the offsprings produced is round. So if this round is self-crossed together, it produces the round offsprings. No wrinkled offsprings is produced. So this is round. So such an offspring or individual is known as, so this is pure breed. Okay. Similarly, if it is wrinkled, crossed with wrinkled, and all the offsprings produced is wrinkled. So this is a pure breed. Okay. So pure breed means homozygous genotype, means this this can be represented with an R capital, and this can be represented with an R capital, R capital. These two letters represent genotype. Okay. Genotype. And genotype is written with the two letters. Capital letter is for dominant allele. Allele. Okay. So dominant allele is represented with capital letter. And dominant allele expresses whenever it is present. Whenever it is present. Or you can say it expresses in both homozygous and heterozygous condition. So it means that if this is R capital R capital, then the offspring's phenotype will be round, and if it is R capital R small, then even the offspring will be round. So you see, when even single R capital is present, the offsprings will be. And the other allele which is represented with this, small letter is a recessive allele. Recessive allele is represented with small letter, and it does not express when other allele is present, when the dominant allele is present. So it will only does not express when dominant allele is present. Or you can say it does not express in heterozygous genotype or heterozygous condition. So it only expresses in homozygous condition or genotype. So this would be an R small or small. So that would be wrinkled only. Okay. So in this case, we decided that the offsprings produced in F1 is all round. So round is dominant and wrinkled is recessive. And because these both are pure breed, so we will write R capital, and we will write R small. This should be a labeled genetic diagram. So these are the parents. This is phenotype of the parents, and this is genotype of the parents. So these are maybe parents P1. Then gamete formation takes place. Gamete formation. So you should write gamete here. During gamete formation, the pairs of alleles are separated. The number of chromosomes is reduced to half, and pairs of alleles are separated. Okay, so every gamete will have one of each pair. So as we are talking about one pair only, so this gamete will have all the gametes produced will have an R capital. From one cell, there are 4 gametes produced, and all these 4 gametes would be of, R capital, R capital, R capital, and R capital. But as all of them are of the same type, so they would not affect the ratios. So we do not write all of them. Instead, we just write the type of, there is just one type of these gametes. Okay, so R capital here, and we write R small here. So they fuse together, and the F1 produced will have R capital, R small. This is known as a monohybrid. Mono stands for single and hybrid means different. So this receives one different allele from each parent. Okay, so that's why it is a monohybrid. And F1 ratio is 100%, round. Now, if this round is self-crossed or self-pollinated, the offsprings produced will be F2. So let's see, this is P2, we have round seeds, which is cross-pollinated with round seeds, as it is the F1 being self-crossed, which is monohybrid. So we will write their genotype. So this is genotype, and this is phenotype. Okay, the gametes produced will be of two types. Half of them having R capital, we put comma in between, and other half having R small. Similarly, the other parent will also have R capital, put comma or space in between, so that it represents that these are two types of gametes, two different gametes produced. Okay. Now to predict the F2 ratio, F2, this is called second filial generation. Second filial generation. Okay. The ratio of F2 is predicted by offsprings ratio, obviously, is predicted by making a Punnett square. Punnett square, or you may say checkerboard. Checkerboard. Okay. So how do we make a checkerboard? As there are two gametes of each parent, so we write gametes of one parent on one side here, and gametes of the other parent on the other side, that's here. Okay. So these are gametes, maybe of male gametes here, or female gametes here. So these are gametes. Okay. Now fusion of these gametes takes place. How do we write their fusion? We should write one from here, which is an R capital, and other from here. Gametes are circled, but genotypes is not circled. And similarly, we should write R capital from here and R small from here. The same way, we prefer to write capital letter first, but that doesn't matter even if you write small letter first. R capital and R small, and here R small, R small. So these are the four possible genotypes. And what will be the phenotypes of all of them? This would be around. Why? Because both alleles are capital or dominant. This will also be round. This will also be round. And this will be wrinkled. So the F2 ratios produced, F2 ratios will be genotype ratio is written as 1 R capital is to 2 R capital R small is to 1 R small R small. But phenotype ratio is written as 3 round to 1 wrinkled. So in this case, you see, one of the parental characteristic expresses itself in F1, and the F2 ratio is 3:1. So a monohybrid cross which produces 3:1, and only one of the parental characteristic expresses itself in F1 is a complete dominance. Okay. Now let's see a test cross. Test cross. As we have understood from this cross that wrinkled seeds plant will always have only one possible genotype, which is r small. Okay, so this is possible genotype. So wrinkles have 100% r small genotype, and we are 100% confident that the wrinkle has r small genotype. However, a round has two possibilities. Okay, a round may be R capital, or maybe R capital R small. So if we have a plant with round seeds, we will be only 50% sure of its genotype. Okay. So how we determine what is the genotype of a round plant? So this is, for example, a round plant, a plant with round seeds, which is with an unknown genotype. Unknown genotype. And we want to know the genotype of this round seeded plant. What we do is we cross it with another plant which has wrinkled seeds. Okay. So for wrinkled seeds, we know that it must be r small. Okay. So when we cross them, the offsprings produced have two cases. Case one is that all the offsprings produced is with round seeds. So it means that neither of the offsprings produced have wrinkled seeds. So it means that this round parent with an unknown genotype is homozygous for, this is homozygous for the round genotype. Okay. As it is a round, for sure we know that one of the allelic pair must be r small. What is the other one? If the other one is r small, at least fewer of the offspring produced should have inherited this r small. So as there is no even a single individual produced in this offspring with the wrinkled seeds, so it means that it is likely that other one is an r capital. Okay. But if case two is, this is case one. Case two is if the offspring produced have plants with wrinkled seeds. Okay, so wrinkle is only possible when it is homozygous for that. So it has two recessive alleles. It means it receives one recessive allele from one parent and other recessive allele from the other parent. So a parent with the dominant phenotype, which is round, must have an R capital, R small. Otherwise, this cannot have the offsprings produced with wrinkled phenotype. Okay, so that's how we can determine the genotype in an individual. Such a cross of an individual of an unknown genotype with a homozygous recessive is known as a test cross. Okay, so how do we define a test cross? A cross or a genetic cross between an individual of an unknown genotype with an other individual homozygous recessive for that character or for that phenotype. Okay, so that's a test cross. Now let's see codominance. This is also known as multiple allele or allelic inheritance. An example of codominance is the inheritance of AB blood group. Blood group has four possible phenotypes, and there are three alleles found in a population for blood group. This is represented with an I. I stands for aminoglobulin, and A is an antigen A. So the genotype is written as IA. The other allele is IB, and the third is IO. An individual can have two of these three alleles only. Why? Because an individual receives one allele from one parent and other allele from other parent. So an individual can have an IA, or can have an IAIO, or can have an IAIB, or can have an IBIB and IBIO and IOIO. So these are possible combinations that are found in a population. An individual can have either of these possibilities, as one allele comes from one parent, other from another parent. If the genotype is IAIA, the phenotype will be blood group A. Even in this IAIO, IA is dominant over IO. So this would be group A. This is codominant. An individual with this IAIB will have a phenotype blood group AB, and this will have group B, and this also has group B, and this has group O. So IO is recessive to both IA and IB, whereas IA and IB are codominant. So let's see. If one parent with blood group A is married to another person with blood group B, okay, all the children born to this couple will be with blood group AB. Okay, so you see both parental characteristics have been expressed in this F1 produced. So this is an F1 produced, as both of these parents are purebred or homozygous. So, and this is codominance, we will write genotype of an IAIA as it is homozygous, and this B, IB, IB. Okay. So gametes produced will be, all gametes produced in this will be an IA type, and in this it will be IB. So when fertilization takes place, these two gametes fuse together and the offsprings produced will have an IA, IB. Okay, so this is an F1. All offsprings produced will have blood group AB. Both parental characteristics are expressing in this monohybrid. This is monohybrid. Okay, so both parental characteristics are expressing in it, so that's why it is codominance. Now, if it is self-crossed means an individual with AB blood group is married to another individual with blood group AB. So this is group AB is married to another individual with group AB. So this has genotype IA, IB, and this has an IA and IB. Okay. So this is the gametes produced will be two types, half of them having IA and half of them having IB. And similarly, the gametes produced in this individual will be half of them IA and half of them IB. Now to predict the offspring produced in the F2, we will make the checkerboard. Okay, F2 ratios will be predicted by making similarly a checkerboard. So this is an IA here, this is an IB here, and we will have an IA here and an IB here. So the possible offsprings produced will be IA here, IA IB here, and IA IB here, and this will be an IB and I. Got it? So this would be group A. This is group A. This is also codominant, and this is group AB like parent. This is also like parents, and this is group AB, and this is group B. So the F2 produced has the same genotype and phenotype ratio. The genotype ratio is 1 IA 2 IA IB, and 1 IB IB. 1 homozygous for blood group A, 2 heterozygous for blood group AB, and 1 homozygous for B blood group. And the phenotype ratio will be 1 group A, 2 group AB, and 1 group B. So do you see here the F2 ratio produced is different from the complete dominance? Okay. In complete dominance, the F2 ratio produced is 3:1. Whereas in codominance, the F2 ratio produced is 1:1. If we look at the inheritance of O blood group, O is recessive to both other alleles. So if a parent with the blood group A, heterozygous for blood group A, is married to a person with blood group B, heterozygous for B. So heterozygous for A will be IA and IO, and heterozygous for B will be IB, IO. So what will be the possible offsprings produced? We will have to make the gametes first, which will be, an IA, other half will be an IO, and here you will have an IB and IO. Okay, so you can predict the offspring ratios by making checkerboard, and this will be an IA, an IO, this is an IB, and an IO. Okay, so this will be an IA, IB, this will be an IB, IO, and here you will have an IA, IO, and this last will be an IO, IO. So this would be group AB, group B, group A, and group O. All the 4 possible offsprings produced if the 2 parents heterozygous for different blood groups are married together. So all 4 possible groups will be produced in their offspring. So this is an F2 ratio. Okay, so this is homozygous recessive. Now let's see sex-linked inheritance. As we know, there are 23 pairs of chromosomes in human, and we can arrange them We can arrange them in pairs from longest to the shortest. Okay. So there are 23 pairs. This is pair 1. This is pair 2. This is pair 3. This is pair 4, pair 5, 6, 7, 8, and so up to 23 pairs. These 22 pairs are always homologous, okay, and are known as autosomes. Whereas this last pair may be heterozygous, as it is seen here. X is longer than Y, so this is heterologous in males. And it is homologous in females and is represented with an X, X homologous in women. Okay, so this last 23rd pair, which may or may not be homologous, is known as sex chromosomes. The inheritance of characters that have their gene on X chromosomes is known as sex-linked inheritance, as X chromosome also has, this is an X chromosome, for example, X chromosome. So there are genes present on it. Gene for color blindness and gene for hemophilia are found on an X chromosome, and inheritance of these two characters is known as sex-linked inheritance. The other characters that have their genes on these other 22 pairs of chromosomes, other than sex chromosomes, is known as autosomal inheritance. Autosomal inheritance. Okay, like eye color, even the blood group that we have discussed, they are present on other than these X chromosomes. So the inheritance of blood group is known as autosomal inheritance. Okay, so inheritance of sex-linked characters gives a different ratios of the reciprocal crosses than the autosomal characters. We'll discuss what does the reciprocal crosses mean. Okay, so let's see. Inheritance of hemophilia. Hemophilia is due to a recessive sex-linked inheritance. Like alleles are found on sex chromosomes or on X chromosome. Gene found on X chromosome. Okay, so a person with X, H capital, as a normal allele which controls synthesis of or formation of a protein. This is called factor 8 gene. Okay, and factor 8 protein produced, which is a normal protein involved in blood clotting. Whereas a person with recessive allele, which is an XH small, has a faulty or mutated allele, or this is a recessive allele, and this one is a dominant allele. Okay, so this recessive allele produces a faulty factor 8 protein, and so blood fails to clot. And as a consequence, a person dies due to excessive bleeding upon even minor injuries. Okay, so a woman can have an XH capital and XH capital. So this would be a normal women, and women can also have an XH capital and XH small. This is a normal woman, but as she has a recessive allele, so she is known as a carrier. And this would be an XH small and XH small. So this is, recessive, homozygous recessive, and this is hemophiliac women. So these are 3 possible genotypes in the women and 2 possible phenotypes. In men, there are 2 possibilities. They can have an XH capital Y. This is normal man, and they can have an XH small and Y. This is hemophiliac man. So if a normal man is married to, this is a normal man married to a carrier woman, or a normal woman who is carrier for hemophilia. So we will write their genotypes as XH capital Y and XH capital and XH small. The gametes produced will be 2 types in men, XH capital and Y. And there will be 2 types of gametes even in this women, and X, half of them having an XH capital and other having an XH small. Okay, so how we can predict the offsprings? We write the gametes on one axis, one side, XH capital here and XH small here, and we write XH capital here and Y here. So this would be an XH capital, XH capital, XH capital, XH small, XH capital Y, and XH small Y. So this would be a normal woman, this would be a normal woman, this would be a normal boy, and this would be a hemophiliac boy. Okay, so 25% chances to have hemophilia. But if we have the cross reversed, the woman is hemophiliac and the man, instead of this cross, if we reverse this cross, now the man is hemophiliac hemophiliac man is married to a normal woman, what should happen? So this has an X, H small and Y, and this has an X, H capital and an X, H capital. Okay. So there would be two types of gametes produced here, and there would be just one type of gamete produced here. So if this fertilizes with this, then XH capital and XH small. And if this fertilizes this, XH capital and Y. So neither of the offsprings produced is hemophiliac. So all normal. Okay. So such a cross may be known as a reciprocal cross. Do you see the results of these offsprings of these reciprocal crosses are different? However, in case of autosomal inheritance, the results of the reciprocal crosses are the same. So inheritance of a characteristic which has the ratios of the offsprings of the reciprocal crosses different from each other, such inheritance is known as sex-linked inheritance. So hope you have understood it. So this is all about monohybrid inheritance. We'll discuss some questions on it, and then we'll move on to the dihybrid inheritance.